Tighten it up, rolling.
And we got hard returns on sonar.
Yep, there we go.
Yeah.
That's bottom.
We have bottom.
Okay.
All right, Nav, can you adjust my sonar to 50, please?
Sure.
On Sirius.
Got a lot of particulate in the water.
Yeah.
Okay.
All right, pilot.
Yep.
I'm stopped just because tether's kind of close.
Yep.
So as you sink a little bit, I'll adjust as necessary, but
Definitely
Delta
getting pushed.
15.
There's a strong current starboard to port.
starboard
see
to port.
Yep.
Copy that. I'm going to rotate slightly to center you, if that's all right. Rotating port.
I'm going to get Z bias in. Actually, what we can do is yeah, Roland. If you can get somebody in the video chair, that'd be great. If you need to go.
It's okay, I've got the ET on it now.
Okay, thanks. Trying to find a stable.
Z bias. So
Okay, thanks.
Yeah, it's takes me a while to get that right.
So Nav, what's our general bearing toward waypoint or waypoint 2 then?
It's going to be generally northwest to the end, but I just heard that they gave me new waypoints.
Okay.
So I'm going to put those in, but it's around 318
Okay.
degrees. I have to do some converting, and then I can actually put them in here.
Okay.
So if y'all are good, I can start working on that.
Yeah, why don't we figure out
Yeah.
where we're going here?
Okay, just give me a second.
Okay, stand by
we
all
can
of you
look
on the
generally
line,
toward, I
please.
think you said 318, is that right?
Yes.
Yes. Northwest.
Let's turn toward that direction.
Copy that. And then, yeah, you'll just want to lateral as you do that.
Yep.
And, yeah, I'll just rotate to keep you centered until we get there.
Sounds good.
Some nice fish.
Welcome to the
That's
conference
interesting
calling
terrain,
center.
huh?
At any time
Yeah.
during this conference, please enter your the
Yep,
passcode you are attempting to enter.
Sonar agrees with 315 Nav.
2, 3, 3.
I'll keep laddering into your view.
Yeah, copy that.
Hey Nav, just let me know that 315 according to D2 sonar.
315 is uphill.
Yeah.
Copy that.
So that generally agrees. We'll bring Sirius around and
A lot of fish
Set
down
up.
here today.
I can't hear you.
Sorry. It looks like there might just be a big rock there on the other side. It might go down.
Okay. Okay.
So I'm looking at 290.
Go ahead, watch lead. Yep. No problem. We're still getting set up here anyway, so. That's totally fine. We're going to do a white balance and get adjusted in our bearing and
Okay, pilots.
get used to the currents. So that's great. It's fine.
We're gonna do it a little bit differently. We're gonna drop targets. So just give us a moment, and we'll have some actually set targets for you.
Yeah, pilot, yeah, if you're ready, if you feel safe, we can white balance.
Let's white balance.
I'm not moving.
I've got autos in, so.
Yeah. Let's see. Video, are you ready if I set it for white balance?
Go ahead. Just go ahead, Sean.
Yes. Enabling HPU, ramping up.
Including you.
When you
Full
went
power
over
pack is
the
enabled.
dive plan, you were quite inaudible.
Shilling is live.
Well, I didn't change anything, so I don't know why that is. It's a mystery to me too. I've noticed the same thing. Sometimes the volume is high and sometimes it's low, and I'm not doing anything different here.
That's weird. Alright, sorry. I thought I enabled valve pack. I did not. All right, Schilling's live.
Yeah, usually we can hear each other on shore on the conference call just fine. At that time, I was listening to the video stream, and that's where you were hard to hear later on.
Okay, yeah.
Okay, we have, looks like we have the phone line restored.
Hi, Shirley.
Hi.
I can hear you.
Good morning, everyone. Roland, I wonder if you could go ahead and give your intro one more time. I know you did it about a half an hour ago, but if you could do it again, I'd appreciate it.
Pilot, can I take your main HD?
Sure.
Thank you.
Okay.
Just going to give it a nice tilt up.
Okay, this is the Okeanos video engineer, Roland Bryan, making a general announcement to all those on the OKEX party line and the conference call. Keep in mind as we proceed with the dive today that all audio is recorded with our archived video, as well as being transmitted across the satellite and out to the world via the internet. So keep that in mind as we proceed to the dive today, and enjoy the dive. Thank you.
Thank you, Roland. Good morning, everyone, or good afternoon or good evening, depending on where you are located. This is the NOAA ship Okeanos Explorer, America's ship for ocean exploration, and we are coming to you live today from Ifuku Seamount, and we are exploring unknown areas of the Marianas Trench National Monument. We have my name is Shirley Pomponi. Sorry, we're getting an announcement.
Yeah,
My name-
if you want to come
my name
in,
is Shirley
and I will
Pomponi.
center.
I am
And,
one of two science leads on board the Okeanos Explorer. I'm the biology science lead. I am from Harbor Branch Oceanographic Institute of Florida Atlantic University and
please center
NOAA's
over.
Cooperative Institute for Ocean Exploration Research and Technology. And our
Coming
geology
up.
science lead is Patty Fryer from the University of Hawaii, and she is
Down.
at a meeting right now in another area of the ship, so she will
Just a
give
little
a
bit
more
further
complete
out than
introduction
normal.
of
That's
herself
maximum
when
zoom.
she
So
comes
this is a
into
little
the control
further
room.
out. Okay, I can get
In
closer if you
addition
want.
to
Or
our
Yeah,
the two
bring
scientists that we
bring
have
the
on
arm
board the
in
ship,
okay,
we have a
can you
science
come out there? 'Cause
crew.
when I
The
bring
rest
it
of
in,
our
it's gonna
science
push
crew
it
is
over
shore-based
too.
at various locations around
So
the
I need to
world,
actually.
either in exploration command centers, in their offices, or even in their homes, and they round out our science team.
Let's see.
My expertise is in sponges, and Patty's is in rocks, and especially mud volcanoes. So we have a multidisciplinary team of scientists who are filling out the disciplines on this expedition. And what I would like to do now is have them each introduce themselves. For those of you who are on the phone line, I'd like to have you introduce yourself, tell us your affiliation,
when I come
and
up,
just a very brief background
All right, tell
about
me-
your expertise.
I will rotate.
How about we start with Bill Chadwick? And Bill, can you please speak a little bit more, a little bit louder than you have been?
Slight nudge down.
Okay,
That looks
this
good.
is Bill
Good.
Chadwick. I'm at Oregon State
Video
University,
levels.
and NOAA's
Interesting, it's hot in the middle.
Environmental
Hot
Laboratory
in
Newport, Oregon.
middle.
I'm
Let
a
me
geologist,
try
And I study volcanoes,
this.
mostly submarine volcanoes.
And we're going to come back to Bill in a few
Did I do
minutes because
anything?
he's going to give us an
right,
overview
brought
of
up
this
the total
dive.
level.
Okay. Chris Kelly.
Hi, I'm Chris Kelly. I'm the
You're
program
brighter than that,
biologist
right?
for the Hawaii Undersea
It's
Research
still hot in the middle,
Laboratory
right?
here at the University
It's
of
hotter in the
Hawaii,
middle, but the total
serving
level
as
is
the
low.
science advisor
Yeah.
for the 3-year capstone project for
So
the Office of
that's-
Ocean
let me
Exploration and
try upper
Research.
swing
What is
arms.
capstone, Bill? Chris?
Interesting that it's so hot.
Oh,
Like
you
there
would
was a
ask.
shadow or
Let's
something
see.
in the middle.
Wait, I can tell you what it is.
Monument.
It's campaign to right.
More level there.
Campaign
those
to
are the
address the
uppers
Pacific
pointed down
Monument
at it
Science,
now.
Technology, and
Does that
Ocean
help?
needs.
It does.
And it's designed to address the need of resource managers to better understand the deepest parts of our nation's marine monuments. I have that information right in front of me,
Yeah, I'm gonna
Chris.
check the blacks.
All right, copy that.
Okay, Bruce Munday.
Exactly as I said it, wasn't it?
It was Bruce Munday.
Yes, I'm Bruce Mundy. I'm a fishery biologist with the NOAA Pacific Islands Fisheries Science Center. I'm located at the NOAA Regional Center in Honolulu, Hawaii, and I'm here to try to identify fishes like the little lantern fishes that we saw swimming around the ROV a few minutes ago.
Yep, Bruce, we rely on you a lot since neither one of us here on the ship have any expertise at all in fishes. So thanks very much. We saw quite a few of them yesterday. And actually, if you're looking through the screen from the feed from the Sirius, you can see lots of stuff in the water. I'm not sure how many of those are fish and how many are zooplankton, but there's lots of stuff in the water. Let's see. How about Tara Harmer-Luke?
I want to cap again and do a black balance, which is...
Hi, this is Tara Harmer-Luke,
Yep, copy
and
that.
I am a biologist who studies deep-sea hydrothermal vents.
It's the flares.
I am an associate professor of biology at Stockton University.
Thanks, Tara. And how, Scott, are you on the
There's a
line or
lot
just
of
in the chat
stuff
room?
Swimming around.
I am on the line, Shirley. Hi, this is Scott France. I am a deep-sea biologist at the University of Louisiana at Lafayette, and my specialty in the last decade or so is deep-sea octocorals, and particularly the genetics of those corals.
Thanks, Scott. Let's see. How about Debbie? Is Debbie Blaney on the line? Okay, looks like she is not. It looks like Ben Frabel is in the chat room. Ben, are you on the line? Okay, and Asako Matsumoto is in the chat room, and she is a deep-sea biologist from the Chiba Institute of Technology in Japan. She's been a regular participant, part of our science team on this leg. So thanks again for joining us on this. And let's see, how about we round this out by having Bill Clancy introduce himself. And Bill, tell us about what it is that you're doing.
Yes, I'm a cognitive scientist at the Institute of Human and Machine Cognition, and I've been working with Harbor Branch Oceanographic Institute to understand how our collaborative tools help us to work together as a distributed science team. And I'm doing a study this week at the University of Hawaii, ECC. So I'm happy to be here. Thank you.
Thanks, Bill. And that gives me the opportunity now to go in a little bit more detail while we're waiting for the. In fact, what you're seeing right now in front of the screens is we're doing a white and color balance on with our cameras to make sure that we are bringing you the absolutely best imagery that we can. The way we operate on this expedition is kind of unique. It's quite a bit different from the way most of us are used to going to sea. What most of us have been used to doing in our careers is getting some funding for a project that we're interested in, and maybe one or several scientists, but we have a crew. Sometimes can be as many as 20 or 30 people, usually not that many, on a research vessel. And what happens at sea is happening within that smaller group of scientists who are participating in that mission. What we're able to do through this type of exploration, telepresence-enabled ocean exploration, is to be able to have a much larger group of scientists, experts, resource managers involved in the expedition. And so what's going on at any point in time, everyone can see in real time, everyone can contribute to in real time. You might wonder how this mission was planned. This was part of, as Chris and I mentioned earlier, this is part of a 3-year campaign focused on, in particular, on our national marine monuments in deeper waters. And for this campaign, it's in the Pacific Ocean. And as we do this, we are partnering with a number of NOAA programs and other partners as well. So NOAA's Office of Ocean Exploration and Research designed the mission and is supporting the mission. Our ship, the Okeanos Explorer, is operated by NOAA's Office of Marine and Aviation Operations. Also involved in this expedition and the effort are NOAA's National Marine Fisheries Service, NOAA's Marine National Monuments Program, NOAA's Deep Sea Coral Research and Technology Program, and another important partnership is with NOAA's National Center for Environmental Information. Through that particular partnership, all of the data that we're collecting
Hey,
are
pilot, do you
being
want
made
your
publicly
sonar changed
available
into 30 meters or
within
anything?
60 to 90 days after the data are
Keep it
collected.
50
So
for
this
now.
is really,
Okay.
really fast for data to be made available to the public. And by the public, I mean
All right,
any of
all
you
good
who
video?
are
Video
watching
clear,
from
thank
home,
you.
as
All right,
well
let's get
as
it
our
stowed.
scientific participants who can then make use
Okay.
of the data to develop hypotheses, write proposals, write scientific
So-
publications. So this is all as a result of very close collaborations and partnerships. In addition to that, we, during our dives, can collect up to 4 samples, 2 biological samples, 2 geological samples on each dive. These samples are also collected on behalf of the scientific community and will be deposited in archival
Okay.
institutions and
I
made
got a
available
whole
to
bunch of
anyone
new
who
waypoints for
submits
you.
a request
All right.
to use and study
Sounds-
those samples. So the
I'll give you an
geological
overview since we
samples
got a little bit of
will be
Yes.
Okay.
going
Please
to
do.
So here we go. We're
to
starting here,
Oregon
and these
State
are completely
University,
arbitrary.
where they will be
Basically, once you get
archived.
past 3,
The biological
so up here makes
samples will
sense.
be going to
Going
the
on top of the hill.
Smithsonian Institution,
And
and
then
in
we're going to
addition
go
to
down
that, we are
the other
taking
side,
smaller
waypoint
samples
3, and then waypoint
most of
4
our
just
biological
arbitrarily at
samples
this nose
to
here.
do molecular
5, 6,
analyses, DNA
7.
analyses. These samples are going to the Ocean Genome Legacy
Why?
8.
at
Okay.
Northeastern
9.
University. And again, these samples
Okay.
are
So general
will be,
trend of uphill,
publicly
but I
available.
don't-
So they're not my
I don't know
samples,
how
they're not
important
Patty's
like this
samples,
guy
they're not
is, and
Chris's
this
samples.
transit here.
Anyone who
Okay.
submits a request and wants to use these samples, studies
It sounds
these
like we're just
samples, may do
exploring.
so.
The entire.
Huh.
That's what we do.
Yeah.
It's like in our name.
So you heard from
Well, I guess we're doing that.
you heard
Let's explore.
introductions
We explore
of
the
a number
ocean,
of
isn't it?
scientists who are on the call this
All right,
morning
pilot.
and who are
come down a little
participating in
bit
this expedition.
So your first
more.
waypoint
and
is
to-
roughly in
from day to day, those
312.
participants may change based on their
Okay.
particular interest in a dive site. The
All right. Well,
particular
we're just going to kind of
sites
follow this
that
ridge
we
up.
have
Yeah.
So they're
chosen
generally
were
312.
based
Okay,
on
but we'll
input
looks like
from the
Sirius has a
scientific community. So in general,
pretty good
the plan
uphill
was put
return
forward
of,
to
yeah, like
conduct
260.
exploration
But I'm just going to
operations
return the lights real
along,
quick.
the Marianas
2 is
Arc,
200
and,
meters away, by the way.
The scientific
Oh, it
community
is.
was
Yeah.
invited to
Okay.
submit
Yeah, so let's follow the local
proposals for particular dive sites,
bathymetry.
and then these were discussed among the scientific
that was a
community.
difficult map
They
to,
were prioritized based on the
contour there.
priorities of the capstone
without any
mission,
points.
and the dive sites were
All
selected.
right. Are we all stowed, Sean?
The dive sites for
yep, that was the
today
last
and
slide.
tomorrow
Everybody's clear?
were
Video's clear?
selected,
Clear.
were proposed
Video
by
rolling?
Bill Chadwick. And Bill,
Video's ready.
Did you get
I'm
the best
going
white
to ask you
balance
if you can give us
gotten in a
a
while?
brief overview of,
No.
of
Just
first of all,
acceptable.
why you proposed the site
Ah, it
and
hurts.
what we expect
I
to
thought with all
see.
that
And Bill, I'm going to ask you
extra
one
time
more
we had
time
today,
if you can get really close
would
to
have
your
fine-tuned it
microphone and
perfectly.
speak
Yeah, I
up
didn't
louder
feel like we
because
had
we're
that extra time.
I don't know about the rest of the group, but
We felt
we're
the
having
pressure to
problems on the
hurry.
ship here.
Okay,
Oh, no.
my final setup is.
All right, watch lead. This is pilot.
Okay, thanks, Shirley. We are at Isuzu
Just letting you know that we're all set up.
which is
We're
one of
going to head to the bottom here, and we're ready to
volcanoes in the
roll when you are.
Mariana Volcanic Arc. So that's a chain of active volcanoes that extends over about 600 miles. or
Okay. I'll just start
1,000
looking around.
Kilometers.
Wait for your word.
There's,
All right.
Most
Oh,
of the
cliff.
volcanoes are submarine, so their tops are below sea level.
Perfect.
There's maybe 50 or
Bridge,
60
RV nav.
of those. And, looks like we're on the bottom.
All right, good visual on
I'll
tether.
keep this
Yep.
straight. There's
Yes, this is.
Yeah, I am seeing some
Bridge, RV nav.
Or maybe you were tugging. I can't remember.
So their tops are above sea level.
Go ahead, Ned.
My motor's just.
Hey,
not
we're on
going.
the bottom, and I just want to give you a
Do you want to come down
temporary
some?
bailout plan
Delta 20.
of 110.
Remember that our
110I. Thank you.
remember that our tether is a few meters longer now.
That's true too. So
About 10 years ago, but we happened to be
a
here
little more play.
2 years ago with a ship, and we decided to look again, and we did see a hydrothermal plume over the summit, and so this is our first opportunity.
We don't want that. Let's zoom in this fish.
Look for that
Hold.
hydrothermal vent site.
That's a nice
that
fish.
has never been seen before. So this is, as far as we know, this is the first ROV dive at this amount, and we'll be looking for the source of that hydrothermal plume on today's dive.
Okay, thanks very much, Bill. And as Bill noted, and as you can all see, we are on the bottom. We are at 495 meters, and we are ready to begin our dive. What will happen during this dive, for those of you who are watching from home, is that from time to time you will hear our shore-based scientists talk on our conference line, our conference call line, and explain what it is that we're seeing. They are also typing in some comments in a chat room, which you can't see, but we will be communicating with each other throughout the dive. Okay, so I've been seeing a lot of conversations going back and forth on the event log.
Ooh, there's something in there.
My first impression is that these lavas look pretty young.
Terrain's very different than what we've seen.
Very different.
It's almost as if it crumbled and collapsed or something.
It does kind of look like that, huh?
Yeah, it kind of looks like a construction zone after a building demo.
Long it takes these corals to grow.
Wow.
Okay, go ahead and zoom.
Yeah, Bill, that's an accurate statement, and I was actually going to say that I'm not seeing very much sessile fauna, those organisms that are attached to the bottom. We are looking at a coral coming up here, but there's not all that much, so that would back up what you said, that this is relatively fresh. This is an octocoral, though, so that's a nice start.
It is indeed. I don't think we saw any on yesterday's dive, did we, Scott?
Okay, holding there.
No octocorals yesterday, just the stylastra and hydrocoral.
And they were really, really abundant.
Sorry, Roland. The vehicle's bouncing as the Z-bias.
Bruce, it looked like there was a lot of fish fauna as well. I saw a lot of exclamation points in the chat room. Did you see something that? Actually, before we do that, Scott, can you tell us what this is? Do you know what the octocoral this is?
That jagged rock.
I'm not certain, but I suspect it probably belongs to the family Plexauridae, which is one of the largest of the common Gorgonian sea fans. You can see it's quite flexible.
And we're seeing some hydroids, right, in the picture as well, those smaller pinnate, feathery type organisms. I said hydroids, did I mean?
Bruce, can you comment on the Randolixes?
No, I don't want to interrupt the coral discussion. We can get back to that. So let me know if it's a good time or not.
Yeah, let's wait until we...
go ahead, Bruce.
Yeah, go ahead, Bruce.
Okay, let's zoom out.
Okay, the large fish that we saw swimming around that was kind of a greenish-orange color is a Randall snapper, Randolixes. It's related to the Onaga and the Ahu, the commercially important bottom fish, and it's also taken commercially. However, these Randall snappers are much less common.
No, there's that jagged rock sticking out.
Yeah, it's kind of tough
Ahu
to...
and Onaga, but that's definitely one of the bottom fish, even though it's not one of the most important ones that we're looking for fishery surveys at the shallow seamount summit. The other interesting thing that's going on in this area is that there's a lot of small fish.
General direction,
and they're
310.
vertically
Right now,
migrating
Sirius is looking at
open water
like 270, and that looks pretty decent.
Fish, the small fish with dots
Sirius
on the
looks
belly
good at
and
270.
sides that produce light that are
why don't we make a short hop for the first one?
Yes.
Yeah.
We've
Let's do
also
20
seen
meters.
a couple of
20
dragonfish,
meters
the elongate micro predators that are
270.
part of that mesopelagic fauna.
Well, actually, just
And Ben
instructed
Travel, I
us
think,
to
said that he
get
saw
moving.
some
So
hatchet fish, same fish.
270.
Yep.
These would be fish
Okay.
that live deep
22
during the
knots.
day.
Hey, can
and
you move your
up
microphone up a
into
little bit?
the
I
more
can't hear you,
shallow
actually.
water at night,
Me?
have a vertical
there we go.
migration. They
Okay.
create the sound
How am I
scattering
nav?
layer that
You're
picks up
fine.
on sonars. When those fish happen to drift over a seamount summit,
Bridge,
like the one
RV
we're
nav.
on now, and they start to descend during the day, the migration is blocked.
Hey, we're ready for a
Seamount
ship move.
summit.
We're going
And
to go
that's probably what's going
20
on
meters,
here. They're
270
accumulating
degrees,
of the
0.2
summit
knots.
of the seamount because they weren't able to complete their vertical migration.
Ship move 20 meters, bearing
However, there are
270
species
degrees.
of these lantern
Do you feel my tongue at all?
Speed 0
that
to
do
2.
little bit.
live
Mine just looks
naturally
pretty.
Yeah,
along
I'm
these
coming
slopes
back.
of
Tight.
islands
Yeah, it's fine.
seamount
I just wanna make
during
sure.
the day and then move up at night. And
You know
could be
that
looking
you're
at
getting some
those
tug from me.
species. There's
Thank
no
you.
way of telling because identification of the lanternfish and other midwater fishes is usually done by looking
So
at the
just help me
numbers,
keep a very close eye
Initiated nav.
configuration
on obstacles
of
and
the
sonar.
light-producing organs. And you
Yeah.
really
Tether
need
hazards.
specimens and even a microscope.
Copy that.
to do
Good
that.
delta.
So we can get
About
the
20.
family on those, but we can't get the genera and species. So this is a great start for the dive. We've seen lanternfish, dragonfish, hatchetfish, and then the Randall snapper. And I think there were 2 of them in the area that I caught a glimpse of. So it's looking like it could be a really good fish dive.
Great, thanks, Bruce.
So Chadwick, I'm noticing in camera 2 there's like tons of things swimming around. Are those all fish?
Yes, those would be the myctophids. I can't say that they're all fish. There may be some shrimps and other things, crustaceans, and maybe even little squid mixed in. But with my eye for fish, I've been seeing a lot of the lanternfish and a few
We're
of the others.
discussing what's in Sirius.
Yeah, this is amazing. This bodes well for the midwater
Can we maybe
dive that's going to
attempt
happen
identify
at the end of the benthic dive.
Or Sirius, whatever these small swimming creatures are.
We can zoom in.
Yeah, if you.
No, and I do
I
suspect
don't
there
think
are
I'll be able to
crustaceans
focus on them, but
and even a little squid
feel free to zoom video if you want.
Well, let's keep Sirius wide,
Okay.
and we can zoom on the ROV on D2.
So I want to make just a brief comment on why I put
Zoom in tighter on
exclamation
D2.
points on that Randalichthys in the chat room. So we're at 496
shrimp.
meters, close to
Right.
500 meters,
shrimp are
and
cool.
currently the bottomfish EFH essential fish habitat designation
What those other
goes
little
to
guys
400
called?
meters,
Copepods?
and I do not believe Randall's snapper
little bitty
is
food
one of
looking
the
things.
management unit
Are these
species
too big?
in that designation.
Okay.
And so there's a couple things. If we see a lot of these, the one of the things we're trying to do here is to understand bottomfish and CNMI a little
Okay,
bit
zoom
better.
out.
So we'll have to see how the rest of this dive goes, but we may be finding some very,
Ship is
very
over there.
valuable
I'm just gonna
that could
grab
be
as I can.
used for the next essential
Okay,
fish
yep.
habitat review.
To have them
So that
consider
looks like terrain shift
that
is
there are these bottom
towards
fish that
your
are current
starboard
deeper
pilot.
outside of their
Yep.
management area.
That's right. We're just doing a short hop.
Correct, and possibly, depending on how many Randall's snappers we see,
Only about
whether
9
or not
meters left
that
on this
ought to
move.
be
Copy that
considered
now.
a management unit species,
I don't
because it
think Sirius
certainly
is
is
gonna
a
catch
commercially
up there.
valuable fish, but it's just in relatively low
I can put up
abundance,
second
certainly
moving if
in
you wanted
Hawaii.
to.
It's pretty rare.
After you go and
What are
check
the
that,
managed
just to get a video.
species?
Yeah, so what you're looking at,
The managed fish
300.
species right now.
So to put me in Sirius spot,
So the onaga, that long-tailed
I'm
red
gonna rotate
fish
to the
that we've seen a
starboard.
couple times
Try to
is one
center
of
you.
them.
That's fine.
There's
That'll actually get me
another
behind,
redfish
get
called
Sirius
the
behind
Azu.
D2, so that'll
We
be
saw
fine.
this on leg
Yeah.
1. I don't
And then we
think
can
we've
start
seen
heading
any
up
on
at,
leg
like,
3 so far. It's small,
like 300.
smaller fish near the bottom. Those
It's
groupers are
280.
management unit species for sure that we saw
A
300.
couple days ago.
days
So
ago.
right there will probably get you to Sirius, yeah?
And the
Yeah, so I'm
striped
looking
yellow
at
and white striped
290 or
one, the
so.
Gindai,
295,
that's one of them as well. So there's a
290,
bunch that we haven't
295. Yeah,
seen
290.
yet that are part of
I'll
the
just hop you
species
up to what,
complex
about 20 meters?
here. But it's just,
Yeah, with it.
I'm
Yeah,
delighted. I
yeah.
just didn't expect to see bottomfish on this dive, and so all this information is really good.
Yep. Bill, can you tell us what we're
Bridge,
saying
RV
here?
nav.
Go ahead, Nav.
Hey, can we put another move in of 20 meters at
Come down a few
290?
meters, give me some
0.2
tether.
knots.
Copy that. Yeah, Delta.
20 meters, bearing
23
290, speed 0.2.
Good copy.
A little bit of starboard to port current. Having to stay about 30% lat to maintain.
All right, Delta's back to 20.
I forget. And what we're going to do
All right.
is traverse.
Yeah, on high pack it actually matches up. That
Great.
you're going to see a little cliff here to go up to 490. That's
Okay.
not too far away.
Right in front of us, we're seeing these are
Cool. All right, let's zoom in.
lavas that
We have
were
10 meter
erupted.
resolution on high pack.
Okay.
As I mentioned at the beginning, they look
Maybe we'll go up to this one.
young.
Go ahead,
They do
Roland.
have corals.
Nah, bridge
Okay.
initiated.
So I'm guessing that takes, 100 years maybe, or decades. So it's not like this came out yesterday, but the lava's very dark and.
It's like he's looking for an escape path to get
Yep.
away from you.
Really more looks
It's
like
fast.
they're
Let's zoom out.
Hundreds of years, so maybe these are decades old lavas.
Looks like they're hunting through
All
the
right,
cracks.
going
to turn bridge down.
Mm-hmm.
Okay, EX up.
Oh, I can't even hear them.
All right. All right, watch leads. We're
Okay, thanks.
underway.
Barely
Waiting
heard that, but
for your word to look
heard
at
you say,
things.
2 or 3 decades old, probably for the corals. Yeah.
Yeah, thanks, Scott. I actually did not hear that. You
Two
want
different corals there.
to, Scott? Should we take a closer look at this coral coming
On
up?
the right coral by your skid.
Yes, please.
Yeah. Okay.
On the right.
Pilot, watch lead.
Go ahead, watch lead.
Yeah, let's take a look at that the coral on the left, please.
Coral
Actually,
on
we can
the
do.
left.
Actually, we can do both. The one
do-
that's dead center that you're.
We can do
you're-
both, yep.
Okay, good.
Possible sponge at the bottom of the screen.
Okay, go ahead.
Be
Zoom
great
in.
if we could look at both of them.
Yep, that's what we're
I'm
doing.
really excited about that one on the left.
Yeah, I thought you might be. Okay, what are we looking at here?
It's got like a bluish
I just
color.
need to wait for the zoom.
Oh, look how the polyps are different. They actually have a
This
thing on
may,
the tip.
so this is an octocoral, likely, although not necessarily,
Hold on.
but likely the family
There we
like
go.
Sauridae. This could be something like what we saw. I'm not 100% certain. It makes it different. But you can see that purple color. This is an animal. It's colonial, made up of several polyps. That's what you see
Captain, zoom.
arising from those
That's
purple
maximum.
stems.
Okay.
You see the rings of tentacles.
We'll
back
make a
in
lighting
the page
change
tells us an
here.
octocoral. Each of those has a mouth.
We
in the
could
middle.
bring
They
out
can
the lower
feed
string arms
and all
just at like a 20-degree
one
angle,
another, but
Sean.
they did all arise from a
I
single
can
egg.
do that.
I don't believe we've seen
Enabling
anything like this
HPU.
on any
I
of our
just had to
dives
take the
so
gain
far.
out.
So I
I
would
got
say
more
this is a
detail
new
now.
octocoral
Still
for
want
the
light bars
Yes,
up?
please.
Oh
Yeah, fuck
wow.
it.
So do we think it might be a new species, new record of
Ooh.
occurrence, or not sure?
How far left in that move? Nav?
We got
They're
4
gonna
meters
pull out
left.
for full species.
Okay.
Little tilt down.
Hold on. Getting there.
Okay, and just a reminder, shore-based scientists, that we have the ability to, an option to collect 4 samples. We have a fairly long
And
dive,
you get
probably
lasers?
another 7 hours.
Lasers are
2 of those samples
coming
are
on.
typically rocks, and 2 biological
rock.
samples. So just keep that in mind, and in terms of our sampling
A long dive
priorities.
today, right? 1830.
Yes.
All right, zoom out. They want to look at this one.
Pilot, your light bars are slightly out.
Yep, that's perfect.
Yeah, that's right.
To see like valve packs. HPU off.
So this
Cool.
is Bob Stern. This looks like a dome. That vertical structure on the igneous rocks make it look like a dome, and I wonder if a sample here wouldn't be worthwhile.
Yeah, I'm
Okay, go ahead, Roland.
nothing looks particularly easy to pick up right here, but I think as soon as we see something that perhaps already
there.
broken off and of
Oop,
a suitable
spinning.
size, we should grab it. I was thinking it kind of looked like almost a tumulus or some like
RV nav, just move completed.
extrusion that cracked
We
down
are
the
there.
hole.
Let's come up a bit, Sean.
Maybe from inflation
Copy that.
or something like that.
Delta 20 and
Okay.
coming up.
Scott,
Oh, were you
What are we
running the
looking at
camera
here?
Or my position?
Your serious.
Okay, yeah.
So we
I
are
think
looking
you
at
copy.
another octocoral here, so the same major group as the purple one we just saw. But here you see the polyps are much smaller, they're slender, thin, and they arise in what we call whorls, these little circlets, and it looks like there are maybe 3. This coral would be in the family Primnoidae, which is one of the most common and diverse families in the deep sea. We've seen many primnoids. Chris Kelly is suggesting maybe this belongs to the genus Calyptrophora.
This is turning out to be a very, very interesting dive already. Seeing a lot of diversities. Something for everyone. Okay, so we had a suggestion from Bob Stern. Hi, Bob. That we might want to collect if we see a small loose rock in and around this area that we might want to collect it. Bill, did you second that request?
Yeah, I think, from what I've seen so far,
I'm not sure what you're talking about that.
pretty massive and
right, it's on that polyp that's on the view.
Okay.
There's something on it, and I don't know if I can.
Okay, roger that.
Go ahead, watch it. Zoom out a bit.
So there's a request from the geologist ashore if we see any of these rocks that are small, loose.
Okay. I will keep an eye out. But if you see anything, you should let me know as well.
Okay, geologist ashore. I've gone ahead and asked the pilot to be on the lookout for some loose, small rocks. But if you see one that you'd like to have, please let us know.
The ship is stationary, by the way, pilot.
Great.
Yeah, to right in this
Yeah,
area.
Sirius is not moving.
It's a bright, shiny fish. Went off screen bottom left.
Looks like there's a lot of loose rock up here.
Yeah, looks like there's quite a bit of loose rock that we're heading towards right now.
Yeah, you can see the striation marks in that. That is neat.
Oh yeah, it's definitely
Oh, yeah.
broken up right ahead.
Well, take your pick, Watchley.
Yeah.
Is Patty here? She's not.
Boulders in the foreground have striations on them that are very interesting. Like I think another indication.
Alright, I'm going to
that
rotate slightly to center you.
That's fine. We're not moving. So.
Yeah.
Okay.
Just 10 degrees.
Okay, so Bill and Bob, you have your pick here. We've got tons of rocks, literally. Which one would you like?
I'd just go for something that looks relatively dark.
Relatively dark.
Stained.
Something relatively dark, not stained. Okay.
I think the stuff on the right there.
Maybe on the lower right.
Yeah.
I agree. Anything is fine.
Yeah, looks like you're getting something right below the laser dots. Might be suitable. Yeah, Bob and Bill, there's a rock just to the right of the
You wanna zoom
laser
in,
dots.
Roland?
If you can
Let's see if that's
see,
loose.
how's that look to you?
It all looks loose.
Hold there.
That'd be great.
The one that the laser's on now? Or just above it?
Well, yeah, the one just above
That
it.
smaller one. Okay, let's
I
zoom
would like
out.
to see you collect that one that the laser dots are on right now. Okay. Okay, so we are going to go in and collect the rock that is a little bit below the laser dots right now. Just for reference, those of you watching, those laser dots are 10 centimeters apart, so about 4 inches apart. And once we finish this collection, we are going to introduce another very important partner, our ROV team and videographers are from the Global Foundation for Ocean Exploration.
Of course. Thank you, Shirley.
So we'll do that, and I'm not going to ask you to. Well, we could have you multitask and
well,
go ahead and
hydraulics up.
you could. You could go ahead and introduce yourselves. And I'm sorry. Well, you were kind of getting yourselves.
Yeah, it was
in place before.
A tricky launch today with all the currents that we're seeing. So.
Yep. So we'll go ahead, and as soon as you're comfortable, you can go ahead and introduce all the guys and gals from the Global Foundation for Ocean Exploration.
Is it the chair? Yeah, sure. Yeah, so here we are, front row. We've got Andy Lister on Navigator. Did a great job this morning with some tricky launch situations. Dan Rogers on pilot, and we have Sean Kennison on co-pilot. And of course, Roland Bryan, video engineer. There's his hand on camera. And
usually Annie, usually our video clippers are Annie and Tara, and she just stepped out for a second.
Yep.
Okay, and again, these folks are all from the Global Foundation for Ocean Exploration, important partner here.
We didn't actually hear them introduce themselves. We must have them on.
Okay, let's bring Mini Zeus up.
You did not hear them. Okay.
Take control, help me out.
Yeah, the introductions were done on the EXRV.
All right, I have your Mini Zeus. Do you want me to kind of rotate over to see craft?
Yeah.
Rotating
we'll
over.
have to do it again.
Okay. Oh,
they're going to go in and
OKX.
get this.
Yep. Is the starboard rock box okay with this? It doesn't look like it's going to be very fragile.
Yeah,
I
it's
figure
fine.
we should save the port one for a more fragile sample.
Yeah,
so.
that's fine.
Okay. Hydraulics coming on. Do a grip force 6. Force back off. All right.
Do you want me to follow you out?
Yeah, sure. And actually, if you want to bring the starboard wing inboard.
Yes, I'll do wing first. Rotating.
That's fine, right there.
For camera or wing?
Everything's fine.
Okay. I am off stick. Yep. Nice and loose.
Look at those poor manip cams working beautifully.
Yeah, it's nice to have the HD image back.
You made that look
You want
easy.
to put it in front of them and spin it to show off to the camera?
Demo it to the main camera or no?
Do you want to look at it in the camera, or can I just put it in the box watch lead?
Yeah, let's take a look at it.
Okay, we can do that.
Since you made this look so easy.
Go ahead, Roland. Get it out more in the light pool. How's that? Anything of interest there? What's that thing?
A little tube. See all the crystals that the geologist has spoke about.
Little red thing on top. What's that?
No,
a little red thing on top.
A little brittle star.
Yeah, it's a little brittle star.
Something on the bottom. Oh, and also on the left-hand edge.
Sorry, I can't.
Okay, that's.
Yep, there you go.
That's good. Thanks.
Okay, zoom out.
Another tube.
That's good. Yep, we saw a little serpulid tube on the rock. We have a few more scientists on shore who have joined us since the dive has started. Andrea Quattrini and Tina Molitsova.
Thank you.
Who else?
And there goes the wing.
Okay, indexed out.
All right.
Actually, hold on. I'm going to test the jaw real
Bruce,
quick.
is that an eel up there that we're seeing?
Seems much more responsive today.
Yes, it is.
Yeah, I was gonna
There's
say.
another one of these
Settings
synaptobranchid eels, similar to the ones
So
we
the
see
thought
in
is that when we had the
water, but
craft
it's in a
controller
different
turned on, the button
subfamily
was
and
slightly
genus,
depressed, and we
this
weren't
group
getting the full range
called the arrow
of sensitivity.
That actually makes a lot of
We think
sense.
this one with the white borders on
I am
the
disabling valve
fin
packs.
It's kind of like
is
having an
medium
N64
to
controller
south
turn on,
name.
you
But
know,
it's one of the more
box with
shallow
the stick
dwelling
depressed
of the
and
synaptobranchid eels. And this is interesting.
Game over.
game over.
It's moving around in and out of the crevices
Hydraulics
in the
off.
rocks
I will
and all.
reset mini
We usually
Zeus,
see these
center
on
it down.
flatter terrain, and often in association with
And
large
then we can
corals,
probably
like
get a
gold
move
corals
in.
or some of the bamboo corals. So
I have you at
suspect that we'd see some large
50 in my
corals
camera.
on this side.
Completely
It's
stationary.
a little bit of an
I think it was
unusual
310
observation.
looked pretty good, didn't it?
I think, yeah, what we're facing now.
Facing
Yeah,
310?
310.
Okay.
Yeah, looks
It
great.
agrees with everything that
Let's go and
we
do,
have.
Let's go and do 20 meters.
Yeah.
You said it's a little steeper, right?
Yep.
Okay.
Yeah, we'll do 20.
All right, watch lead. We're going to get a ship move in and keep moving uphill.
Okay, roger that. Okay, folks, we're going to be continuing
Bridge,
to
RV
move
nav.
uphill. We are currently at 498 meters.
Hey, another ship move. 20 meters, 310 degrees, 0.2 knots.
Back up, please.
Working on it.
That looks pretty good.
What bearing?
And Sean, if you can range down on
310.
D2 sonar now, that'd be great, to 30 meters.
Yes, of
I think
course.
Is it this
Oh,
one?
is it that mouse? No, I think
No, it's the other one. Yeah, you're right. I switched them, so. All right.
Just waiting for a response. There we go. So you want 30 meters?
Yeah, 30-meter range is great. Thank you.
Oh,
This is
here
move
we go.
initiated.
All right.
All right, Roland, you see anything you want to look at?
Maybe that's staining
Okay,
on the
there's
rock.
Okay.
Yeah, Bill or Bob, can you talk to us a little bit about the different the striations and the staining that we're seeing on these rocks? What that means?
Moving closer for you, Roland.
I think we could probably get it from the float.
Oh, so much faith. That's good.
Total faith.
Okay.
Z bias.
Go ahead and zoom in.
Move down slope a little bit. Any
Hold
kind
there.
of yellow or red staining is probably when the rocks were.
There's like a brittle star that's
and there was a
camouflaged there. You see him?
water flowing
Yep.
through them that reacted to with the iron in the rock. That's what that stain basically kind of like rust.
Mm-hmm.
The iron they get
Zoom in
altered
more.
a little bit.
There's a
turn those
tube
colors.
off to the left with a fan sticking out of the top
But this
of it.
looks like the lavas were
That's
very
cool.
were pretty viscous as they came out. The striations are probably either there were big pillows that have fallen apart, or maybe even bigger flow low.
So, pilot, does that mean we're going to have to recalibrate the grip forces?
We're seeing a beautiful brittle star there on the left
I
side
think
of the screen. And if you move a little bit more to the left, that looked like there was something else. Yeah, really. Okay, there are those white,
Sponges at
the white
top.
spots. Are they?
Go ahead and zoom
What
in.
are they?
Maximum. It
Are they
does look like
sponges?
sponge. It's got
They
little
look like
holes
they
in it.
might be sponges. Very lacy looking.
Nowhere is a.
Okay, and
Four,
then as you pull out up to the right, not that far. Oh yeah, actually, okay, up at about 2 o'clock now.
2 o'clock.
Yeah, that looked like something reddish on the rocks, circular.
Up above, I see it.
Yeah, you see it's coming in the center of the screen. Let's
Yep.
see what that is. It might just be stain, more of
Go
this,
ahead, zoom in.
the
With the
iron stain,
shadow.
but I'm not sure. Yeah, okay, so just more of the stain on the rock. And what do we have over on the far right? Is
Like a little squat lobster?
Squat lobster? Looks like a little squat lobster.
Okay, zoom in. Hold there. Yep.
Yeah, Scott France said that when we were looking at that ophiuroid, that brittle star looked more like a carving in the rock rather than a brittle star that was sitting on the rock.
Okay, zoom out.
Where are we seeing? Tina is saying a stalked crinoid. I do not see a stalked crinoid.
Right where the lasers are?
You see it, Ron?
Right where the
It's a very,
lasers
very
are. Real? Oh my gosh!
Off to the left
Really?
Yes.
Let's. Yeah. Can we get in closer to that?
Hold there.
I'm
It is.
not sure about that.
Yep. It's got the rough stalk and the crinoid arms at the top.
Okay, let's zoom out and get a little closer. Set down.
Yeah. So these. Yeah, we're gonna get a little bit closer so you can get a closer look at that.
How are we doing in that move?
Oh, we're done.
We're done? Okay.
Yeah.
Cool.
I
Perfect.
have you at 65 in my cam
That's fine.
pilot, so you still have time.
Yeah, that's fine. Okay, go ahead.
One next to the brittle star.
I don't think that was.
Right
Is that
hand
it?
side.
Yeah.
Right there.
That's I was looking at the one in the middle.
Yeah, it's
Looks
over
like
to the.
same thing.
Go to the right.
These little
Yeah.
Up. Right next. Yeah. Go up. 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 There we go.
max zoom.
Oop.
Tiny, tiny, tiny. Really tiny. Okay, that is probably the tiniest stalked crinoid I have ever seen.
Okay.
Okay, and Scott thinks that as tiny as that is, that he may have even seen some amphipods on there. So, wow, really tiny. Okay.
Do you want to look at this shrimp watch lead?
Sure.
Okay.
Yes, please.
Zoom in.
I think he's the generic long-legged one, but
Beautiful shrimp.
Oop.
Les Watling, are you on the phone line?
max
If so, do you want to call in and talk about this
Go
crinoid?
backward. There we go.
you are zoomed into something that's only a half-inch long.
Yep. It's crazy because I feel like I'm bouncing around a lot, and then I look at the manipulator cams, and I'm not really moving at all.
Okay, well, we're seeing a
Just zoomed in.
shrimp right now.
It felt great. Josh seemed to be working fine. Had full range
Tina
of sensitivity.
Molosova from the P.P. Shirshov Institute in Moscow says that some of these types of, some comatulate crinoids, so those are the unstalked ones, have stalked larvae, but these seem to be adults. So right now the thinking is that these are just tiny stalked crinoids.
Yeah, so
I'd love to
I'm
get a
done
screen
moving.
grab of that
I
and
have
send
been,
it
so
to
if push out a little. We could probably
Chuck Messing
get
at
another move
Nova.
in. You can do
Yeah.
another
Objects
20 meters
laser,
if you
right?
want.
Yeah, I think we
Okay.
could probably do. Yeah, we'll get that. Yeah, 310 still looks good to you.
Yeah, we can get past.
Yeah, once I push out a bit, it's a pretty steep here. So
Yeah.
So something on the right-hand side.
Should flatten up ahead a little
Okay.
bit. Quite a bit, actually.
Fuzzy and
So we can
white.
do maybe one more short move and then go from there.
Yep.
At about
You
2
wanna look
o'clock,
20 right now?
Ron? Is that what you're looking
It's
at?
more 5 o'clock now.
Let him push out
Oh.
a little more.
Bottom edge.
Oh,
See what
yeah.
you're saying on the bottom of the rock.
Yeah, bottom of the
Yeah.
rock.
Just in case they
Like
take
it
a
might
long
be a
walk.
Let's just hold here for
Coral.
now, but we'll get the move ready.
Okay. Okay. In the lasers right now.
Okay, go ahead.
Oh, pretty. Another octocoral.
There we go.
Looks like it might be similar to the second one that we saw.
On the main stalk, you can sort of
Yeah,
see scales.
beautiful. Okay, yeah.
Coming out. Coming down.
So some of the,
there we go.
yeah, oh, beautiful imagery. Really beautiful. See that, the polyp on the, almost all the way over on the left, not the one, yeah, like where it's just bent. Is there a crustacean?
Is that full zoom?
It's full
That's
zoom.
full, okay.
And I can't get
Yeah.
any closer.
I
Yeah.
think that maybe it's, I don't know, it's something in the center.
Shirley,
of
I'm wondering if that may have eaten something and
Yeah.
stuff like that. I mean, that's the position where the mouth is.
Right.
You know, there's something.
Right, that's what I'm thinking too, Scott. It looks like there was something that that it had either just eaten or was eating right now. Scott, can you say something about how we identify corals? Because you said you have no idea on this one. What are some of the characteristics we use to identify octocorals?
Sure. So we would look at the arrangement of branches and the arrangement of polyps, and what you can see here is that the polyps themselves, where the tentacles are kind of transparent, and they seem to be
Something off to the
emerging
top
out
left.
of a cup. So that's one
See it,
characteristic that we
manipulator
can use.
cam.
But really, what we have to
What is
look
that?
at are there are skeletal elements, little pieces of calcium carbonate that take on a huge myriad of shapes. They can be pointed needles, they can be little balls, they can be like jacks, all kinds of different shapes and sizes, and we have to look at those in our microscope, and I'll let you go and talk about the urchin.
Okay, thanks. Okay, we're seeing a beautiful urchin here.
Zoom in, and I'll move a little closer. Drift closer.
Lots of barnacles.
Hold.
And as we saw the other day, well, we'll wait. I wonder if Chris Ma from the Smithsonian is going to call in. But Chris was telling us the other day about these barnacles that, oh my gosh, you can see the barnacles that have their cirri extended. So you can see those little barnacles that are attached to the spines. And I believe, if I remember correctly, when Chris Ma from the Smithsonian called in the other day, he said that this area, the spines, of course, don't have any tissue on them, and so the barnacles can attach to them. And I believe he said, correct me if I'm wrong, or maybe Scott, you can fill in some of the details, that there are some that are very specific. So there are specific
Instead of,
barnacles that are attached to specific
pushing my
urchins.
toe into it, I'm going to get on top of it here.
Yeah, I'm afraid I don't know enough about the barnacles, but I believe that there are some barnacles that are host-specific. That's all I can say.
Yep.
Zoom back in.
Thanks. Yeah, that was my recollection, that they were host-specific.
Heel in the background.
Zoom in. Hold there.
And you can also see kind of on the lower left, actually, if you look all the way around, but the really long tube feet in between the spines on this urchin. So the urchin is probably a deposit feeder. It's grazing along the bottom, and it's got along for the ride these barnacles that have their beautiful feeding tentacles or cirri extended, and they're probably capturing particles that are in the water column. Beautiful.
Cool. All right.
Okay, that's good.
Zoom out.
Yep, that's good.
Some really interesting rock formations
Yes,
here.
very, very interesting.
Let's look at this thing on the bottom left. What is that? Snap zoom?
I think it's a shadow.
There's more staining. Oh, a little brittle star.
These
Those
These
red and white
brittle
ones.
stars are incredible. They do look like rock paintings.
Okay, let's zoom out. I think I see a big coral fan up above this. This is a cool formation. Just keep an eye on that tether, huh?
Yeah. No, copy that. Delta.
Okay, we have another
18.
octocoral.
Come up a meter.
Wow, they're
You
just
wanna
like
look at the one on the left or the right?
pick one.
Let's
We'll
do
go
the
to
right
the other
one.
one next.
The right
We'll go
one
to the
looks
right
different.
one. Yeah, we'll go to the right one first.
2 different fish at the top of the screen.
Okay, zoom in, Roland. Oh. I will get a better angle. Hold on.
Partial.
Yep. My toe's almost down. There it is. Can you zoom in more?
Beautiful, beautiful.